Hello everyone, welcome to Gateway. In this video, we will discuss about the PYQs of GATE regarding the topic recombinant DNA technology. So, let's move to the first question. The first question is the type of nucleic acid present in alpha lambda phage is A. Double-stranded DNA, B. Single-stranded circular DNA, C. Single-stranded DNA, and D. Single-stranded RNA. So as we know, lambda phage is a virus and it has single linear double-stranded DNA of approximate length of 49 kilo base pair. And also one important point to be noted here is it has cos sites or cos ends, which is sticky in nature, right? So these are some points to be noted. So we have the answer here. The correct answer is option A, which is double-stranded DNA. Okay, let's move to the next question. The recognition sequence of four type 2 restriction enzymes are given below. So the symbol arrow, this arrow indicates the cleavage site. Identify the restriction enzyme that generates sticky ends. So we have to identify those restriction enzymes which generate sticky ends. So let's first make the complementary sequence of these restriction sequences. Okay. So G A T C 3 dash to 5 dash CTG CAG 3 dash, and this is 5 dash CCC GGG 3 dash and 5 dash AG C and T. Let's start with the fourth one, this one, last one. So the restriction site or the site of cleavage is two bases away from the 5 dash. So in the complementary site or in the complementary sequence also, the cleavage site will be two bases away from the 5 dash end. So it will be here. So the cut will be generated here and it will result in a blunt end. Similarly, for the third one, the cleavage site is three steps or three bases away from 5 dash. So in the complementary sequence, it will come here and the cut will be generated like this. Also a blunt end, 5 dash end, three bases away, 5 dash end, three bases away, also a blunt end. So the answer is obvious, first one, but still we will see it. 5 dash end one place, so the cut will be generated here and the end will be like this, GATCC 3 dash and 5 dash G. So it is a sticky end. I hope it's clear. Let's move to the next question. The next question says a circular plasmid has three different but unique restriction sites for enzymes A, B, and C. So there is a plasmid and it plasmid has three unique restriction sites for enzymes A, B, and C. Okay. When enzymes A and B are used together, two fragments of equal sizes are generated. Equal size are generated basically. So this is the plasmid, and if we use enzyme A and B simultaneously, it is generating two fragments of equal size. So if the site for A is here, the site for B should be exactly opposite, right? Consider it as a 2 kilo base pair plasmid. So there will be two fragments generated due to these two cuts, right? There will be a left fragment which is 1 kb and there will be a right fragment which is also 1 kb. So there are two fragments of equal size. Okay. Enzyme C creates fragment of equal size from only one of the fragments generated by those cleaved by A and B. So after cleavage of A and B, two fragments are generated and the enzyme C creates two equal size from any one of the fragments, either from the left one or right one, but the fragments generated by C are also equal. So the site of cleavage should be somewhere midway, right? Consider it on the right side. Okay. So it will generate, if it was 1 kb and it was also 1 kb fragment, so After the cleavage with C, the fragment size will be 0.5 kb and 0.5 kb. So in this plasmid, if we will see the cleavage sites, so it is the site of A, it is B, and it will be the C site. Okay. This fragment is of 1 kb, this is of 0.5 kb, and this is of 0.5 kb. I hope it's clear till now. Next, the plasmid is treated with a mixture of A, B, C and analyzed by agarose gel electrophoresis. If we apply all three enzymes simultaneously and analyze it by agarose gel electrophoresis, what is the number of bands observed in the gel? Okay, when we apply all the three enzymes simultaneously, this is the result. So, if we see the agarose gel bands, if it is 2 kb, 1 kb, 1 and 0.5 kb, so after the treatment with mixture of all the three enzymes, there are three fragments generated. Three fragments. One is of 1 kb and there are two fragments of 0.5 kb. Right? So there will be one fragment or one band at 1 kb and one band at 0.5 kb. So the answer is two bands. I hope it is clear. So the correct answer is 2. Okay, let's move to the next one. Introduction of foreign genes into plant cells can be carried out using A. Agrobacterium, B. CSL2 mediated plasmid uptake, C. Electroporation, and D. Gene gun. So there is a difference between plant cell and animal cell. Plant cell has a cell wall protecting it, and inside it is a plasma membrane. But there is no cell wall in animal cell, directly plasma membrane, right? This cell wall confers or provides rigidity or stability to the plant cell, right? Now, the first one, agrobacterium. We know it is used for introduction of foreign genes in dicot plants. It is very obvious, and this question is of MSQ type. Okay, so the first one is correct. Second, CSCL2 mediated plasmid uptake. This method is used for animal cells, right? So this is not correct. And electroporation, third one, electroporation is the use of electricity to generate small or minute pores in the plasma membranes, due to which the foreign genes incorporate. Okay, so electroporation is used for cell wall, for plant cells. Okay, so this will be correct. Also for animal cells also, both, for both of them. Now gene gun. This gene gun is a technique in which it is also called as biolistics. Biolistic. There are minute sized particles of gold or tungsten coated with DNA, and it is bombarded on the cells. Due to the bombardment, this animal cell cannot take up the force and breaks, but these plant cells can withstand that force. So this gene gun is used for incorporation of foreign cells in plant cells also. So the correct answer will be A, C and D. Okay, I hope it is clear. Moving to the next question. Among individuals in human population, minor variations exist in nucleotide sequence of chromosome. That is true. These variations can lead to gain or loss of sites for specific restriction sites. Okay, if those variations occur at the site of restriction enzyme, like the recognition site, it might be lost. So what is the technique used to identify such variations? Okay, so what the question is asking, let me explain it to you. There is a, for example, consider EcoR1 enzyme which recognizes a site GAATTC. It is the recognition site. Okay. Consider a human chromosome on which there are three restriction sites present. Okay. And after the application of this enzyme, it will result into fragments, right? Here it will cleave from this side, this place and this side, right? And it will lead to the production of fragments. This will lead to the production of this fragment, this will give this fragment, this will give this fragment, and this is the fourth fragment. Okay, so there will be four fragments. If any mutation occurs in the place of this specific recognition site, for example, consider it might become GSCTC. So this echo R1 will not be able to recognize this site. So out of these three, consider the middle one has got this case. Okay. So this recognition site of the middle place, second one, is lost. Now if we apply the echo R1, it will cleave on these sites, and which will result in the generation of fragments, only three fragments, and there will be a large single fragment in place of two small fragments. Correct. This is the case. This is the question. And the question is asking which is the technique that identifies this difference. So let's move on to the options. We all know polymerase chain reaction or PCR. This is used for amplification of DNA. So this will not be the answer. RT-PCR, no. So option A or C will be, any one of those will be the correct answer. Let's see option C. Polymorphism. What is polymorphism? Polymorphism is change, difference, basically difference. Length polymorphism. Difference in what? Length. Difference in length. You can see here there are differences in length. Fragment length. Whose length? Fragments length, right? And these fragments are generated from, why those are generated basically? From the use of restriction site. So the use of restriction site due to use of, sorry, restriction enzymes, due to the use of restriction enzymes, there are fragments generated, and those fragments have differences in their length. And these differences are identified by using this technique, which is known as restriction fragment length polymorphism, or RFLP. So the correct answer is option C. I hope it is clear to you. So guys, if you like this video, please do subscribe to the channel. Thank you.